Okay, you should have your cell communication notes, new notes packets. This will cover topics 4.1 through 4.4 based on the unit plan. So here we go. This really is a time to take notes over the content that we learned on the POGL when we were in class this past week. So the POGL introduced us to the idea that cell communication can be broken down into different categories based on how far the signaling molecule or ligand is going to need to travel. And so the first section of your notes would be the category or the types of cell communication where the molecule really doesn't travel a distance. Now, it's a bit of a misnomer because, yes, the ligand moves, but it doesn't move very far. And so here we're going to find two types of communication. The first one is autocrine signaling. Now, we saw that autocrine signaling right here in this top picture. This is when a cell makes and releases its own ligand that binds to a receptor on the cell, either on the membrane or possibly an internal receptor if we're talking about a ligand that's nonpolar. So autocrine signaling, this would be like if you are talking to yourself, if you are communicating with yourself. The example from our POGL, which is a great thing to remember, cancer cells, they actually make and release their own ligand, a growth factor, that triggers the process of mitosis, which is why cancer cells can sort of perpetuate their own cell division. The second type, which technically is classified as a no-distance communication, is our juxtacrine signaling. And so remember, this is when one cell releases a ligand that binds to a neighbor cell, but that neighbor cell has to be in direct contact with the cell next to it. So we actually need physical contact between cells. The POGL talked about these things called plasmodesmata. These are little channels that extend between plant cells that allow for the cytoplasm to stream back and forth from plant cell to plant cell and carry any kind of molecule within the cytoplasm, including ligands. In animal cells, a very similar structure would be called a gap junction. So the important thing here is in juxtacrine signaling, you all of a sudden need two cells in the communication process. So this is not like autocrine, where it's one cell communicating with itself. We have two cells that are talking to each other. Those cells make physical contact to communicate. So those two are no distance types of communication. The next section of your notes talks about short distance communication. So the ligand moves, but it doesn't go very far. And so in short distance, this is our paracrine signaling. And the paracrine signaling, remember, is when one cell releases the ligand, and that ligand will bind to another cell that is in the nearby area. But keep in mind, those cells, the two that are talking to each other, do not touch each other directly. So there's no direct cell-to-cell contact. Here is an example right here of paracrine signaling. This is what happens at the end of one nerve cell. So this is the end of one nerve cell, and this is the beginning of the neighboring nerve cell. And so it's going to pass ligands across this gap that exists between the two of them. And so in our POGL, we learned about neurotransmitters. Those are the ligands that are specific to neuron or nerve cell communication. We also learned about morphogens, which are the molecules or the ligands that help as an embryo is growing, turn cell types into tissue types for the organism. And then we learned about how bacteria communicate with each other, that process called quorum sensing, and that is also paracrine signaling. So 3 really good examples from our POGL. Again, if you didn't do the POGL, you should go back. It had really great examples that we are pulling from. And so if you're wondering, how does a morphogen work? How does quorum sensing work? How does a neurotransmitter work? All of that was explained in the POGIL that we did this week. Now, I'm going to toss in a phrase here in case you see it in a question set on AP Classroom, on the real AP test. It's called synaptic signaling. And so that is another term for paracrine signaling that happens specifically in, it says neurotransmitters. It should say in neurons. So here's where it gets its name. Remember these two cells over here that are going to talk to each other by passing a neurotransmitter across this gap? This gap is called a synapse. And so synaptic signaling is passing a neurotransmitter across the gap from one neuron to a neighboring neuron. So just in case you see that term or phrase synaptic signaling, you know what it is attached to, specifically the nervous system and neurotransmitter communication, which is a type of paracrine signaling. Oh, look, we have our arrows. Lovely. Last section here would be long distance signaling molecules. So this is endocrine signaling. Now, endocrine signaling, we're going to build a little pathway, which is why the arrows are up here. Endocrine signaling starts when one cell releases a ligand. And typically that ligand is released into the bloodstream, into the circulatory system. That ligand then travels by way of the circulatory system to a target cell somewhere else in the body. And so the distance that the signaling molecule can be as far apart from your head to your toe, as long as there is a circulatory system pathway to get it there, it can travel long distances. Once it reaches sort of its target area, the ligand will diffuse out of your circulatory system and bind to a receptor on the target cell. Because the ligand travels a long distance, these are typically pretty durable ligands. And more specifically, we could take a clue from the endocrine signaling back to the endocrine system, which is shown right here in this diagram. The endocrine system is a collection of organs and glands. And so you see these sort of colorful tissues and glands. The key to something that belongs in the endocrine system is going to be a gland or a tissue or an organ that produces a hormone. So if it's making a hormone, it's part of the endocrine system. And as a result, most of the ligands in endocrine signaling are also hormones. And so think about hormones. This is a connection, and we made it on Wednesday in class when we were all together. We made this connection, but in case you missed it, hormones are a type of lipid. They come from steroids, one of those funny ringed fats. And because they are a type of lipid, hormones are nonpolar, which means they don't necessarily have to bind to a receptor on the outside of a cell to start a communication process. Because they are nonpolar, they are invited directly into the cell. And so oftentimes endocrine signaling is happening internally because these hormones really can enter and exit cells pretty easily because they are nonpolar. So receptors for a lot of these endocrine signaling systems are going to be located within the cell, not on the surface of the cell, because of the nature of the ligand. The hormone is nonpolar. The hormone can move into the cell freely, and so it will find its receptor inside the cell more often than not. So here's a quick comparison. I know, it got real grainy. Autocrine signaling, a cell that produces its own signal molecule, binds it back to its surface. Here is juxtacrine signaling. So two cells are touching, but we do see now communication between more than just one cell. There are two cells involved. Paracrine signaling, two cells, but a distance that separates them. And endocrine signaling, two cells that are further apart in that signaling molecule. That ligand is traveling by way of the bloodstream. So all of that was POGIL, and we talked about that in class this week. This is a tiny bit of new information. And so section 2 of your notes is called the signal transduction pathway. And so let's take a few notes here on this big fancy term. This sometimes can be referred to as STP, the signal transduction pathway. And really, it's a generalized path of cell communication. So this is, in general, what happens as the ligand binds to a receptor somewhere and eventually causes some kind of response. So the signal transduction pathway is broken down into 3 steps. And you can see, check out the diagrams, you can see the 3 steps here. And then you can also see the 3 steps, 1, 2, 3, right here. So step number 1 is called reception. Step number 2 is something called transduction. And step number 3 is called the response. We have really focused on so far, we've focused on an understanding of reception. The ligand binds to the receptor and then something happens. The reality is each of these 3 steps needs a little bit more detail, and part 2, step 2, the transduction part of this needs probably the most. And so for the purpose of this lecture, to prepare us for our next class, I'm going to talk about reception right now, and then we'll build into transduction for our next lecture. So just a few more slides and we're almost done. Okay, so some details regarding reception. We know that a ligand binds to a receptor in the reception process. Great name, right? There are a couple of different kinds of receptors. So, namely, what we just talked about with hormones, an intracellular receptor is a receptor. Intra as a prefix means inside of, internal. So this is a receptor that is actually found inside of the cell, not on the surface of the cell. And these intracellular receptors typically bind nonpolar ligands, the ligands like hormones that can freely move across the cell membrane and bind to a receptor inside of the cell. So this picture up here shows us the boundary. Here's our cell membrane, the boundary of the cell, and then a very large nucleus with some DNA in it. All this white is outside of the cell. Normally, we have talked mostly about signal molecules binding to receptors on the surface. But because this signaling molecule is nonpolar, it can enter the cell and it will bind to a receptor inside the cell. Those are called intracellular receptors. The other kind of receptor are called extracellular receptors, and these are the receptors that are on the surface. And so here is a picture of several different kinds of extracellular receptors. These all have different shapes. They are all protein receptors, and they all have a bit of a docking place for the ligand. So the ligands that are going to bind here are the ligands that have a hard time making it across the cell membrane. So really big molecules, polar molecules, and ions. Also, in the process of reception, once a ligand binds, the protein that it bound to changes shape a little bit. This is called a conformational change. It is literally a change in the shape or the conformation, the configuration of the protein. And so it's not just about the ligand binding to the receptor, but it's about the receptor initiating a little bit of a shape change that then triggers the next part of cell communication, the transduction step. So ligand binds, which causes the receptor to change its shape. Here's a quick little animation, and we're not even going to watch it with the audio. It's a little too technical, but I want us to look at and just sort of watch this process. So here is our ligand, and it is coming to bind to this receptor protein. But the protein is not just what we see in yellow. It's all these things that are attached here. Watch it bind. The protein changes shape. It sort of pinched itself here in the middle. And more importantly, it sort of chopped off. It's called cleaving, like a cleaver. It chopped off a part of itself, and that part floated away. So that's an example of a conformational change. That's a pretty drastic change in the shape of the protein. I mean, it chopped part of itself off. But watch it again here. The ligand binds, the protein changes shape, and part of it is removed. And this part actually goes on to cue the transduction step. So it's almost like dominoes, a chain reaction of events when cells are communicating with each other. Okay, so that's enough for right now. We'll save transduction and response for our second lecture, but have this done and ready to go by class.